Great. Well, thanks for including me. It's nice to be part of this important symposium. So I'm new to Stanford, and I got here in September, and the team is just coming together, and it's a diverse team with lots of different disciplines coming together, a lot of different perspectives, and we as a group recognize that diversity is a fundamental tenet of innovation, and we all learn the most from those we have the least in common with, and I think it's appropriate for everyone to know what our values are, and we do this to make sure that people understand that we can be a great destination for excellence wherever it is. And so I want everyone to sort of know that as we get started here. As we think about opportunities in precision health, I think the perspectives that we have, like a lot of people, come from a lot of different viewpoints, and certainly molecular science is front and center for much of what we do, and we bring to that also engineering principles, understandings in biology and clinical translation. And with that perspective, I want to share with you sort of three stories of precision, and starting at a systemic delivery and then going local delivery, both the pancreas and to the skin, the dermis. But when we think about systemic delivery and we think about particles, for us, we're trying to take the tools of the computer industry, the top-down lithographic tools used to pattern silicon for use in all of our memory and computing devices, but using those approaches for making particles that have the precision of size and shape and chemistry. And so what we do is we've been in the past patterning silicon wafers down to 50 nanometer features. We've been coating those wafers with liquid fluoropolymers with a very low surface energy that can wet every nook and cranny of these wafers, and then we can photochemically cure it and make a mold that can be expressed in thousands of linear feet. And then we can bring these molds together with other liquids shown in red that can become the foundation of a particle. As we precisely fill these cavities of predefined shape, we can solidify these liquids in these cavities for them to be particles. And then we can harvest these particles by bringing two films together with an adhesive through a roller and nip in a web-based process, and isolate these particles and dissolve away the adhesive and have colloids. And so we're able to scale this up using this approach and make particles in the tens of milligram scale, and do that with precise size and shape, and becomes really useful then to also control the chemistry and the chemical composition, the porosity, the modulus as well. And these kinds of precision tools have allowed precise deposition into the lung. And a company like Liquidia spun out of our laboratories, a publicly traded company on NASDAQ, and multiple drugs in the clinic for precise size and shape for delivery to the airway. Also thinking about systemic approaches with these same sort of particles, mimicking red blood cells and precision related to modulus, and mimicking the mechanobiology associated with red blood cells, being able to get through fenestrations in the lung or the spleen, but also going into precision of size, getting down to sort of 80 nanometer-like structures to extend the composition of drug or the concentration of drug in plasma. When you inject a small molecule into an IV, it gets cleared in your body very quickly. And so the whole game is, can you extend the concentration time profile of drugs, the pharmacokinetics of the drug, by taking a small molecule like Taxotere, put it into a particle that's got a certain release kinetics, and this can have better outcomes in various animal models, and really trying to precisely tune the concentration of drugs for enhanced efficacy using precision carriers. Now, one of the big challenges in precision medicine is a challenge of delivery. And, a lot of our imaging techniques are getting better and better, and diseases are being found earlier and earlier, but too many of our treatments are still systemic. When the cancers may be local, or maybe they're local and they're not accessible. Pancreatic is really notorious for having stroma that basically acts as a fortress to protect the cancer cells. And these tumors with a blocked lymphatic lymphatics tend to have a very high hydrostatic pressure. And so when you do systemic delivery of drugs, they go everywhere else in the body but the tumor because of these barriers and the high pressure. And so using an engineering approach, we're really interested in how do we access these tumors that are in this situation when they're locally advanced but not resectable in particular because they will compromise some important arteries and veins, the portal vein and the superior mesenteric artery, and that really limits access for surgery. And so we're interested in trying to debulk these kinds of tumors. And knowing that drugs systemically don't get to them, we've developed precision delivery approaches with devices where one has an ionophoretic device that has a reservoir, a semi-permeable membrane. This is an illustration of a cross-section of a pancreas with a tumor. One that we can, through a counter electrode, use iontophoresis and electroosmotic flow to get drugs into some of these tumors that are poorly vascularized. And so this is a device that we reported on a little while back using iontophoresis for local drug delivery. But what we found is that when you can take a look at tumor volume, this is a PDX model with my colleague Jen Ye, former MD PhD student James Byrne, that what we found is when we deliver drugs locally using this ionophoretic approach, compared to systemic treatment, we can actually debulk these tumors. And this is the first time that it's been shown in the literature that one can debulk these PDX models. And we think that really sets us up well for making more people eligible for surgical treatment. And so the company ACT, Advanced Chemotherapy Technologies, was launched based on this at Research Triangle Park, and recently the Vinod Khosla and the team funded that company in RTP to take this into the clinic. Another precision delivery opportunity and need is also thinking about to the dermis. And a lot of us are getting our vaccines. I just completed my two treatment with the Pfizer vaccine. And, it's really interesting that vaccine is going into my arm, into the muscle, and we have far fewer of the immune cells that we're actually targeting in the muscle compared to the dermis. And there's a lot of, it's just a great example of, if you could get the needle, the vaccine to the dermis in a way that's scalable for the population, we'd have a much better approach. And a lot of people, including Mark Prousens and others, have been looking at microneedle delivery. And this is an example, just like the particle technology, where precision for fabrication tools is really needed. The tools for making microneedles have emanated from the microelectronics industry, just like we were trying to do for particles. And these fabrication tools are exquisite, but they can have lots of limitations on the kinds of materials and the kinds of geometries that one has for this. And we've been focused on microneedles that have a lot more latitude related to maybe being solid microneedles that are coated, or maybe hollow microneedles that can deliver liquids through them, or dissolvable microneedles that can have time release characteristics. And the idea that you could have a solid microneedle that might just be a sheet of plastic, so that you could insert that into the skin and leave the molecules in the dermis where you want them. And so we developed an approach using 3D printing that we think gives a lot more latitude in design of microneedles, and a lot more latitude in the chemistry of the microneedles themselves, opening up new opportunities for local and precision delivery. And so just as a quick illustration of something you can see easily with your eye, this is a macroscopic device, happens to be an air vent for an airplane, printed using this 3D printed approach, where you basically have a light coming up from underneath the window, allowing us to now continuously fabricate really complicated unmoldable geometries using a light-based fabrication approach. And this is the kind of thing that we want to adopt for doing this for making microneedles. And so the whole approach for doing something like that is to take this and apply it to a system that allows us to control the flow of liquid underneath using these membranes that are permeable to oxygen, also allows light to pass through, and allows us to continuously fabricate objects using this new approach to 3D printing. And so we've been able to understand this approach in a lot of detail, controlling light and oxygen and the fluid mechanics with this approach. And ultimately, without going into a lot of details, and you can look up some of our references, I think of this as a software-controlled chemical reaction to grow parts. And so what the issue is, now how do we do this at really smaller features that would be relevant for microneedles? And that's controlling the pixel size and the frame rate at which we can make these. And so we've developed one of these printers with really small pixels now. And now you can start seeing geometries that would be really difficult to access using traditional microfabrication technology, even needles of different diameters or different heights or undercuts, and doing it out of a wide range of chemistries is a real challenge. But we can directly print these. You see hydrogel particle needles, you see biodegradable needles, you see water-soluble needles. And this is just an example of some chemistry that we 3D printed that have used polyacrylic acid of different molar masses that we control with the printing process. And with that, they have different dissolution rates. And when you have different dissolution rates, you can attenuate the delivery over different time scales and get into things like a prime plus boost approach or more complicated design strategies. And so what we've been doing is we're beginning to design these needles for local delivery where we use coating operations. We're also making the hollow microneedles, which open up opportunities for interstitial fluid sampling as well as liquid delivery. And so my team for a long time has been interested in vaccines on the particle-based approaches where we do the vaccine kinetics. We inject the particles into the foot pad. We watch the accumulation in the draining lymph nodes. We've even been doing that with RNA delivery, self-amplifying RNA replicons. And what we're now doing is doing this with protein-based vaccines using microneedles. And this approach basically involves a free meniscus coating. So we're basically dip coating individual microneedles into individual wells that contain either antigens or adjuvants, typically in a solution with sucrose or polyethylene glycol or methyl cellulose. And when it dries, it's a solid state solution of these biological cargoes on these microneedles. And not our data, but data from the literature would suggest that this is a great way to mitigate some of the cold chain challenges for delivering biologicals. And what we've been doing then is that we've been using labeling approaches and optical imaging to track the dissolution kinetics from the skin to the draining lymph node. And we've been doing all sorts of analysis of the antibody and the humoral response and comparing it to a subcutaneous injection versus microneedles. And what we see is that there's a tremendous uptick in IgG antigen-specific antibody response relative to a subcutaneous. And so we're getting actually 50-fold more antigen-specific antibody response by delivering the same vaccine, the same amount, to the right place compared to doing a subcutaneous type of injection. So the value of precision delivery is really self-evident there. And so the whole approach now is we think microneedles is really the way to go going forward in a plug-and-play platform approach for delivering whatever stoichiometry and suite of antigens and adjuvants that one wants, to do it in a way that would mitigate or at least ameliorate a lot of the cold chain issues, opportunities for a prime plus boost in a single dose, and be self-administrable. Imagine just in this pandemic, if we had a situation like this where we could all dose ourselves with something like a Band-Aid and leave it on our arms for a couple hours. And so we're putting together an approach. We're working with a great group here at Stanford and University of North Carolina, Mark Davis, Peter Kim, Ron Levy, and others, and then Ralph Barrick and John Smith at VLP Therapeutics, looking at the subunit protein vaccines as well as the mRNA and the self-amplifying RNA approaches for local delivery. And then building the technology allows us to be scalable at high volumes is a key part of this. And the last thing I'll just point to is that beyond infectious diseases, the opportunity in cancer vaccines and being able to prime the antigen presenting cells, the dendritic cells that are poised for developing immunotherapies and others are really key. And we're working at this and come back to that pancreatic approach I started with. We think this is a great way to go into precision medicine. So covered a lot of things, but three examples from systemic delivery to local delivery and the benefits from that going forward. Thanks very much, Ryan.
Excellent. Thanks, Joe. Very exciting stuff. Yes, super hard to change all these things under like normal conditions. I think you bring a level of sophistication I haven't quite seen in microneedles, especially.
Thanks.
So I wonder. So is the mechanism related to the time release as well as the depth of penetration, or what's the are those the driving factors for this, or is also the shape of the microneedles? I'm just curious.
It's a range of things, but first and foremost, we have a larger population of these immune cells in the dermis than we do in our muscle, and the diversity of population of these immune cells are different. And so, the ultimate goal is we've got to get things to the draining lymph node. And the dermis is a great place for that. We have a net of immune cells in our skin, evolved from us cutting ourselves and preventing infection. And it's where the rich opportunity is. And so, again, if you've got molecules that you want to get to a certain spot, you might as well take them there. And that's the whole premise of the microneedles. And then the question is, how do what's an effective way to deliver it? Getting to a very high surface area per needle allows one to get to, high cargo loading per needle. And then by tuning the chemistry, you can get into the all important kinetics of delivery of drainage so that you can get sustained delivery to the lymph nodes so you can get the appropriate depth and breadth of an immune response. And we're actually seeing a more balanced Th1, Th2 immune response. And then it opens up all sorts of, mucosal delivery to the mouth and being able to deliver, microneedles to buccal delivery to the gums of the mouth, and opens up, especially for respiratory diseases, more of a mucosal response. So we're excited about where that's going.
And then I guess, are you also taking into account the pain associated with it? I mean, it depends. Mostly, I think, on the geometry and things like that as well. But maybe you can comment on that.
Yeah, I can't testify directly myself, but regarding microneedles, they say that they're more pain-free than needles. The needles weren't that problematic for most people either, but people do advocate that one of the reasons that microneedles are useful is that they're a pain-free way to deliver things. But I think that's less of an issue. But I think it's an advantage that many point to.
Sure, I would imagine that's probably true. Have you also looked at other contexts like allergy, for instance? And I wonder, in that context, for instance, could you apply? I mean, you want to kind of modulate the immune response to get the immune response, but also so you can do subsequent treatments as well. So have you thought about, because people get I've seen people doing studies where after so many doses, they start to actually see quite a bit of redness and irritation. So subsequent doses might be painful potentially. Just curious what your thoughts on that.
Yeah, well, the reactogenicity that you see with your vaccine is really critical. I mean, that's important to actually see that. If you don't see that means you're not getting much of a response. And so that's important. But, to your point, the delivery to the dermis and to the lymphatic system as if you think of that as a big path forward, and just like delivering via blood is a big path forward with IV, we have 5 times more interstitial fluid than we do blood. And, the lymphatic system is an important system to be delivering through. And I think this gives us access directly for whether it's allergy type systems or cancer vaccines. And we do realize there's some great opportunities in stimulating T cells and we think about, I think it could be an opportunity for doing, CAR T cell therapy, in vivo, without sort of the blood extraction, and could we stimulate T cells directly with targeted particles and access in this way? And so we think there's a lot to go in that direction.